Well, welcome back, everyone. I guess it's going to take a few minutes before all participants come here and enjoy for the second lecture. Okay, so I think it's about the time. So, okay. Okay, so ladies and gentlemen, good morning. Associate Professor Dr. Nuduan Muangsan from Suranari University of Technology, who's serving as a chair for this plenary session. So it's my great honor to introduce Professor Lourdes Cruz to deliver a panelist lecture. Professor Lourdes Cruz is a distinguished Filipino biochemist renowned for her pioneer research on chronotoxins, which is bioactive peptide derived from the venoms of Conus marine snail. Her extensive work has significantly advanced the understanding of peptide, leading to their application as biochemical proof for studying neuron activity. Moreover, her research has facilitated development on omega chronotoxins used for investigating calcium channel in neurons, and mu chronotoxins used for inhibit muscular movement during synaptic studies. So beyond her scientific contributions, Professor Cruz had been instrumental in initiative connecting science and technology to poor and indigenous rural community in Philippines. So her remarkable achievement had earned numerous awards, including being named a national scientist for the Philippines in 2006 and receiving the Royal UNESCO Award for Women in Science in 2010. So please join me in welcoming Professor Dr. Lourdes Cruz to deliver a panelist lecture.
Hello. Okay. So the title of my talk is on chronotoxins. And I want to introduce what these chronotoxins are. They are peptides that we found in the venom of the cone snails, marine cone snails. And so I'll discuss the chemical characterization and the mode of action of these peptides. Oop. Okay, so we got interested on cone snails way back in the 60s. And at that time, there were already studies on the taxonomic and geographic behavior of these diverse species. Alan Kohn was the pioneer to do scientific studies on the conus. And so he studied the habitat of the cone snails and recorded distinct cases that that he gathered. And at the same time, the cone snails were very popular among shell collectors. So I was working right next to, we were not very far from the shopping center of Manila, and there were a lot of shell stores, and we would see these very beautiful cone snails that were being displayed in cabinets. And the story goes that, well, some of them are very expensive, and that they are gathered by the fishermen. And one of the stories they told us was that at one time, the fisherman didn't want to give the species that he was able to pick up from the sea. He didn't want to give it to the owner of the boat, so he kept it under his armpit. So the owner wouldn't see it. And then they said he died because of that. Okay, but that's probably not true. It's just to discourage the fisherman from hiding the very nice species. And so among the references that we looked at was a book by Jerry Walls. It didn't have any publication date, but we were looking at it in the 60s. It was probably published in the 50s. It's still available, and it recorded 500 different species of cone snails. And of these, about 200 are from the Philippines. And in the 1960s, studies on their ecological behaviors, ecological roles, and mechanisms for venom delivery were studied. And these are the 7 deadly cones. According to one literature, these 7 deadly cones are supposed to be analogous to the 7 deadly sins. So, but of these species, we know that, I wonder why this keeps moving. Okay. This one is the most dangerous, the Conus geographus, and that has been recorded. This has been recorded as the most deadly cone snails. According to Yoshiba of Japan, they have the fatality rate is about 68% of untreated venom cases. And they said that these are all deadly, but nothing has been recorded about really verified as to the deadliness of the other cones, although they cause severe effects on people. And the most dangerous ones are the fish hunters, which is Conus geographus, here Conus tulipa, and Conus magus. And those that have what they call triangles on their shells are the mollusk eaters. All of them are mollusk eaters. So when we first gathered these cone snails, we just had one aquarium. We placed them all in one aquarium. And the following day, the species that we liked to experiment on was gone. It was already eaten by Conus textile, so the mollusk hunter of the group. And in the 1970s, the field expanded to biochemical research with the first successful isolation of venom peptides and the growing clinical awareness of cone snail stings. The pharmacological studies was done on crude venom by Endin of Australia. Con also did a little bit of biochemical experiments, but we also did some biochemical experiments. And these are the fatalities, the cone snail sting cases that were recorded in different years. And that's at the time that, well, from 1950s to 99, there was a big increase in the sting cases because probably people were becoming very curious about it. So during the colonial times, these were already gathered by the European colonizers because of the beautiful shells. And they said that at one time, Conus Gloria Maris even outpriced the work, the painting of Vermeer when they had an auction. So it was that the rare species were very valuable. Okay, my work didn't start off with cone snails, but right after graduation, we were recruited by the International Rice Research Institute, for I worked there before and after my PhD. Then I thought, I got tired of working with rice, and I thought one would be more free to work on whatever you want by going to an academic institution, where really there's academic freedom. And so I got my my BS in 1962, my PhD in 1968, and then I went back to the International Rice Research Institute. Then transferred to the College of Medicine, where there were a number of biochemists who just graduated, who just got their PhDs from the United States. In fact, we were just 1 year or two apart from when we were in college. And then for a while, I did some research with Baldomero Oliveira on the DNA ligase and pyridonucleotides. And then we decided that it's so hard for somebody in the Philippines to compete with the researchers in the United States or in the Western world. So we thought of working on something where we will have an advantage, because we have very little facility, we had very little funding, and it's impossible for us to compete with the other researchers abroad. And so we thought, well, we have plenty of cone snails. Why don't we work on this? And they will not be able to compete with us because they don't have the cone snails. So, so that's why we got started on this project. And then I also work with other toxins. Okay, they have a very wide species of prey. Some are worm hunters. Many are worm hunters, in fact. Some are mollusk hunters. That's why Conus textile was able to eat Conus geographus, and so that the species disappeared. And there are about 100 only of fish-eating cone snails. And the total is about 700 different species by that time. And the estimate now is about 1,000 species of cone snails, because they keep finding newer species, particularly in the deeper parts of the ocean. So this is how, there are two types of fish eaters, or even three types. One is that there are the net hunters, and what they do is they open their mouth very wide, You can observe that in the aquarium. And then they, they open their mouth very wide. They'll sting a fish that comes around and then close the mouth and swallow it whole. And then in about 2 to 4 hours, they regurgitate nothing but scales and bones and maybe a harpoon. And so this is called the hook and line fishers, because they use their proboscis, their extended proboscis, to sting the fish and then pull in the fish. So, the fish gets paralyzed, and sometimes they struggle, and so this is how they do it. Okay, and this is how the harpoons look like. For the net hunters, their barb is not very well developed, but for the hook and line fishers, you see that they have a very, very well-defined barb. So the evolution really works very well. I mean, the strategy for catching coincides with what they have, the instruments that they have developed. So Conus geographus is admittedly the most dangerous cone snails, and all the fatalities that have been recorded are due really to Conus geographus. And so Yoshiba said 67% is the fatality rate. And I remember reading about him in that he injected himself with the venom from a worm hunter in Japan, and observed, all the changes that were going on inside his body. A bit crazy. So, and the venom apparatus is this one. It is a radula. The radula is here at the tip of the proboscis. The proboscis can be extended to very long, even far from the snail. And then they have the radula sac, where you have harpoons at different stages of formation. And then you have the venom duct, where the venom is synthesized. And then a muscular bulb, really muscular, but when you dissect it, it really does not have any venom that we can see. And when this extends, they can extend even twice, maybe, for as long as their shells. Okay, so we asked then, what is the nature of the toxins? Is it a carbohydrate, lipid, or protein? And then, how are we going to assay these toxins? And then, so we decided to see if it is a carbohydrate, lipid, or protein. We look at the literature and saw that Alan Kohn has done an experiment trying to digest the toxin with a protease, and he found that there was no decrease in activity. But we wanted to try it ourselves. So we used a higher temperature, longer incubation time, and found that indeed, by treating them with enzyme, like chymotrypsin, trypsin, or a bacterial enzyme, then the activity was lost. And as far as paralysis was concerned, we decided to consult the pharmacologist about 3 floors from where we were. And so we learned how to do injections in mice, intraperitoneal injections in mice. And I said it was so inconvenient trying to watch the mouse for how long? It will take them before they get paralyzed to assess the level of toxin in the sample that we have. So we decided to to hold the screen, to set up a screen, and then place the mouse under the screen, the injected mouse. And then we designated it as the falling time, for the purpose of monitoring the activity. And indeed, if you plotted the reciprocal of dose and the reciprocal of time, it was a straight line. Okay, so we then proceeded to try to separate the toxins we had the cold room of all the old facilities that we had. Then we bought columns, gravity-fed chromatography column. And then at first, we did this by hand. Then, we graduated to a fraction collector. And then we had a monitor that came from the International Science Foundation of Sweden. So these are the principles. For you students, these are the principles that we use for separating the toxins. You can use the molecular sieve to separate them according to size. Then one can use their charges also. And these were the two that we were using at first. And then I found that when you put it in an anion exchanger, you get, the chromatography peaks, and the toxin did not elute till the very end, where the material was very, very low, but the activity was still very high. And that excited us very much because that means that the toxin is very active. But the problem was it was very difficult to tell the amount because I was using only a colorimetric reagent, ninhydrin assay. So we decided to bring this over to the University of Utah, where Toto Oliveira was working, and we collaborated with Bill Gray, who was just a floor above us. And what we did this was a series of the old chemical reaction we did, even separated the toxin by chromatography, but the result was still crude. When you separate them by chromatography, we did high voltage paper electrophoresis, which probably most of you haven't heard of by now. And monitoring was done initially by ninhydrin reaction, Lowry method. And then we did acid hydrolysis. Hydrolysis of the peptides into their component amino acids. And when we saw the pattern of the amino acid profile, we were very excited because not all the amino acids are represented. There was integer ratio of the different amino acids. I think there are about 9 different amino acids out of the 20, because a big protein will have amino acid analysis that will have fractions or non-integer values of the component amino acids. And then at that time, we had to do determination of amino terminal groups. It was by dancylation, and then determination of amino C-terminal end, and trying to determine whether these are blocked or not. And we found that the peptides are blocked at the C-terminal end, with amino, with an amide at the end. Um, by then, there were developments in HPLC, or high pressure liquid chromatography, and particularly we heard that Bill Gray, the one who was helping us, was was trying out then the reverse phase chromatography via HPLC, and we were using the C18. The reverse phase is you have a non-polar stationary phase where different lengths of carbon, alkyl carbons are connected. These are C8, C4, C or whatever length of what do you call it? The carbon groups are attached. And then we also tried the hydrophobic interaction chromatography. And there were also columns that were molecular sieves, Cephadx, and the ion exchangers. So we used all of this in trying to characterize the cone's peptides. Okay. In trying to find out, the most difficult thing is that once we learned how to do the peptide analysis, then we decided to synthesize the peptides. And we were still, at that time, the best one was the spinning cup method of peptide synthesis. And just to synthesize a 22 amino acid long conotoxin, it took me a week of day and night work in the lab to do this. And then we tried to look for, because they cause paralysis, they cause death, maybe a neurophysiologist will be able to help us. It was Doju Yoshikami, also from the same department, that helped us with the initial work. And then later on, we found that Nair and Sackman discovered the patch clamp technique. This is a method for looking at individual ion channel molecules, which is very powerful. You can tell what they actually do. Because before the patch clamp technique, we were looking at ion channels at the membrane only. We use biochemical calculations. We had a course called physical biochemistry. And, it was so abstract. And you didn't appreciate, the value of the channels until they discovered the patch clamp technique, where one has a single cell, you poke electrodes, you pull the glass tubes very finely, and then you poke the cell and dry. The cell, if you touch the electrode on the cell, this will be attached, and then you can either have an inside-out patch where the channel is inside the electrode, or a patch where the channel is outside the electrode, or you can have a whole cell patch for the behavior of more channels. But you can really watch, when you put a toxin from the electrical activity of that single cell, of that single ion channel, whether it's blocked or not. And So we were so excited when we saw the first omega conotoxin. These are calcium channel inhibitors. If you introduce the calcium ions, you can assay for calcium channel inhibitors. If you put in sodium, you can assay sodium channel inhibitors, and so on. So nowadays, we have Oliveira's group has developed constellation chromatography. What does constellation chromatography mean? It means, well, the advantage is, when we started off with assay, so we were using whole animals for assaying the toxins. And then to learn about what they're doing, it's good that we were able, in collaboration with Richard Chen of the Yale University, we were able to see how they affect the ion channels. And his brother, the brother of Richard Chen, was the one who developed the calcium dyes that will have different colors when exposed to calcium. And so what they did is to maybe, they would have different types of cell. At each nerve or muscle, at each nerve, we have different, at each tissue, then we have different types of cells in these tissues. And for this constellation pharmacology, you can assay a number of different molecules with trying to learn more about what they are doing to different types of cells. So the use of pharmacological agents, set of agents, constellation pharmacology is the use of different pharmacological agents, including both agonist and antagonist, to differentiate between cell types according to their specific constellations of key signaling proteins. So if 8 different pharmacological agents were applied to 200 different cells, then you will have, you can generate 1,600 assays already. So it's very fast. And the individual responses of 100 to 200 cells are simultaneously monitored by calcium imaging. So this is the calcium imaging. You see this arrow pointing to this where nothing happens, and then this becomes red with this reagent, and this one becomes red, and so on. So it's really a very powerful technique of trying to facilitate what these toxins do. Because formerly, it was so difficult for us. It took a long, long time for us to determine what they do, what is their mode of action. And so like, for example, if you have a cell class, this may be in the mixture, thermosensors, mechanoreceptors, proprioceptors, pure-receptors, nociceptors, and the sensation that you get with these receptors, you have the temperature, touch, position, itch, and pain. So you have discrete cell types for each type of sensation. And then you have the low threshold and high threshold feeling, when you have that particular cell types. So this can differentiate the different types of cells that's associated with a group, with a particular cell type. And for now, they have found that the pharmacological, particularly, we tend to purify natural products, but for therapy, they're now looking at using a combination of these peptides. Maybe the cone snail were ahead of us they really did excellent pharmacology because each peptide, each venom contains 100 to 200 different peptides, each acting on different ion channel. And the peptide of a particular species is different from the set of peptides in another species. So they are all different. And in a particular tissue, for example, to affect pharmacological change, you may have, if you target only one type of receptor or one type of ion channel, you may produce, you may get an effect, but that's not complete because there are other things in that mixture before they can actually be very effective. So in the cone snails, they use, like Conus geographus, when you inject them peripherally, they have the alpha-conotoxins that targets the acetylcholine receptors. They have the mu-conotoxin that targets the mast cell subtype of sodium channels. And then the calcium inhibitors are the omega toxins that are effective on fish but causes shaking in mice. And they don't really affect people that much. They cannot kill people. They're not involved in the killing of people. But mice, the fish is, of course, their natural target, are very sensitive to them. And when we were first isolating the toxins, you have a number of HPLC, because the resolution is very high, you get a number of HPLC peaks, and maybe up to 200 different peaks for a single venom. And the only ones that showed activity were those that were injected peripherally in the, in the stomach, what they call this, subcutaneous or the intraperitoneal cavity. These are only alpha and mu-conotoxins. And the rest, all the others, maybe be more than 100 of them, had no effect on the mouse. Until an undergraduate student said that, I wonder what the cone snails will do, or what the venom will do if we inject them into the brain. And so he went up to the medical school. We discouraged him, but of course, being an undergrad student, he would just do whatever he wants to do. And he went up to the medical school and learned how to do intracranial injection. And when we saw a variety of activities from these cone snails. One peptide causes running activity. One causes, some jerking, which we named differently. And there was a peptide that causes sleeping in young mice. We isolated this, and this turned out to have a high proportion of gamma carboxyglutamate. It's 17 to 21 amino acid long, and it's different from the other peptides because of the presence of gamma carboxy amino acid residues. And then we call this we named these conotoxins, but the one that causes sleeping, we named it after a Filipino word, and so that's now called conantukin. Antukin in Filipino means to sleep. And then we found one peptide that when you inject intracranially, the mouse doesn't want to move. But if you push it a little bit, it will walk and then settle again, rest again, and we'll do that, several times. So we decided to call it tulakin because tulakin means someone has to be pushed to do something. So we have conantukin, contulakin, and then one of my graduate students found a peptide that causes copious urination, but we decided not to translate that to Filipino anymore. So, one can have fun doing science by attaching little names to all of these peptides. Okay, so in the course of our studies, we collaborated with a number of scientists. We have pharmacologists, of course, working with us. The first advice of the pharmacology just right above us in the College of Medicine was in studying any toxin, you should first do the LD50. Oh, we were shocked because you used 100 mice for that, and we didn't have material. But being chemists, we decided, let's work first on the structure. And after that, we were able to synthesize the peptides, and then we had material to work on. Then we worked with protein chemists, Bill Gray and Jean Riviere, synthetic chemist, Jean Riviere also. He's very famous for his work on hormones. Then the we have molecular biologists also working with us. Dave Hilliard, physiologist, Doju Yoshikami, electrophysiologist, Richard Chen, structural chemist doing NMR, and molecular pharmacologist, where we have some pharmacological agents and seeing how they interact with the different toxins. Then, of course, we started off with very low technology. We didn't have much equipment in the lab, but we were able to gradually buy an HPLC and so on. Now the University of the Philippines has a sequencer, a synthesizer, and mass spectrometer, and NMR. So we have these all developments, essential technology developments that made possible our work on the developments that we did on cone snails. Without them, it would have been impossible to do what we have done. The patch clamp technique for ion channels, and now genomics, transcriptomics, proteomics, and constellation pharmacology are really doing a lot for the advancement of conus peptide research. And you may see this here, that the first model that we did was based on a space filling model. These are balls that you attach to each other to just simulate the structure, that is way back. You probably don't encounter space filling models today. And then came the computer. And so these are computer models of the different conus peptides. And now where it was so difficult to determine, to even synthesize the right conformation of peptides that will be active. And in fact, in one of our researches, we were so excited because we had this very strong activity. And then, but when he synthesized the peptide, it didn't have any activity at all. We were wondering whether we didn't fold it the right way, or is it real? It turned out that the peptide that we isolated was a small impurity in the peak. So one has to be very careful as to say that this is the one that causes activity. You must synthesize it first. Make sure that you get the activity of the native that's found in the native toxin before saying that it really is the peptide of choice. Okay. So we have come a long way from species diversity. We now have, Conus as the apex. Yes, we have now 1,000 different species. That's the last estimate, and more than 200 peptides. The conus, and we consider cone snails as a gold mine of neuropeptides and possible drugs. They are the hidden treasures of the sea. And it turns out that the former estimates of number of conus peptides and molecular diversity is an underestimate. According to work done by Gao et al. Of China, that these are the known species of cone snails. The Philippines, more than 100, Australia, 166, India, 100, China, 60. And that the original estimate was 100 to 200 peptides. And recent integration of genomic, transcriptomic, and proteomic data suggests that this is an underestimate. 100 genes per species and thousands of peptides, because when you synthesize the peptides and look at their pre or pro region, they have different cutting lengths. And then at the same time, there are many post-translational modifications. There are peptides like bromocontrifane that is brominated. Some have sulfates, of course, a lot of hydroxylation. There is a change in hydroxylation of threonine, rather proline, and so on. And then carboxylation is also a post-translational modification. And the cone snails are found in these warmer parts of the world. Most of them are in the Indo-Pacific region with the coral triangle. Let me see. This is red. That means it has the highest number of species, cone snails per species, the red area. So it's in the coral triangle. You have Indonesia, even Thailand, Philippines, a little bit. And in fact, when I went to Bangkok a long time ago, I went to the bookstore and found a novel about the cone snail being used as a weapon to kill somebody. And it was also featured in one of the Hawaii Five-O episodes, that the cone snail was used, cone's textile. But of course, the effect was exaggerated with the cone snail causing the death of the victim just a second after he got stung by the cone snail. So, because that has caught the imagination of people. So right now, there are thousands of peptides, and the current estimate is about 1,000 species of cones. And this is also Fuying Dao. We determined the mode of action of the different sets of, with different cis patterns. We determined their activities. But using AI, he advanced the conotoxin classification, and he did this circular illustration of how many different types of conotoxins there are. And we have families and superfamilies of conotoxins. They affect different ion channels, and they affect also the G-coupled receptors. So they can affect a lot of different physiological processes. So I tell our students that biochemistry is fun. And we enjoy doing research and mentoring students and young scientists, discovering compounds. Of course, there were moments when we got negative results. We were all so depressed. But then it's good working with a group. You collaborate, you tell each other what to do, and someone's depressed, then the other one is not, then you sort of help each other. And our work and scientific publications became recognized and highly cited by other scientists. Actually, when we were doing this, we were just enjoying research. We didn't aspire for any prize. We didn't aspire. We thought it was good to publish. And then, came the awards, which was really a topping to what we're doing. And now, the omega conotoxin M7A is marketed as Prialt. Prialt is used to alleviate pain, intractable pain, but unfortunately, we didn't patent. At that time, when I was a graduate student, being involved in commercial ventures is a big no. But things have changed. Now I'm glad that these have changed. And people are in fact encouraged to invest, to be involved in startup companies and so on. And at the time, the atmosphere at the College of Medicine was not all that encouraging of research. In fact, one of the deans told me, "Oh, you just be thankful that we allow you to do research on your own time." So that's why I transferred to the Marine Science Institute because of that. And so then came the awards, and we were so happy. And so I tell you, chemistry can be fun. So our students here. So it can be a great foundation for graduate work and professional careers. You can go into, as long as you know the basics of chemistry, you know how to prepare reagents, you know how to compute for the buffers, the pH, and that. Basic things that you should learn when you are in high school or in college. So this can be a good preparation for chemical sciences, chemical engineering, material science, medicine, dentistry, food technology, pharmaceutical science and industry, paint industry, agriculture, veterinary medicine, and of course, the biosciences. And one uses math skills, problem-solving skills, skills in scientific thinking, enhance resourcefulness and creativity, appreciation of teamwork, honesty, and ethical values. And then, of course, when you become scientists, you practice professionalism. And I tell you, one of the perks of being scientist is being able to travel around. If you like to travel, if you enjoy traveling, the red dots are the places where I have been to. Of course, in the United States, we're doing a lot of collaboration there. I've been to Africa, Madagascar, South Africa, Kenya, to New Zealand, Australia, and around here in the Southeast Asian region, and even to Russia in Vladivostok, and China and Japan, and then of course a number of cities in Europe. So if you like to travel, that can be a big perk, and you travel for free. So the big question was, for decades, it was a big problem for protein chemists how to predict accurately the folding of a particular protein. So we have the sometimes they tell even the number of different the character of the amino acids that will follow a particular amino acid will depend how it folds. And so, but still, the accuracy was very low. And then in 2024, the Nobel Prize for Chemistry is in proteins. Okay. David Baker succeeded with almost the impossible feat of building entirely new kinds of protein. And Demis Hassabis and John Jumper have developed an AI model to solve a 50-year-old problem predicting proteins' complex structures. I remember having to read Charles Stanford's prediction of how the peptide should fold. So it's a difficult reading for me. But now with the use of AI, I think they use DeepSeek for this. So, you can have a shape that you want, then you can predict what amino acid should be there, and you can build it. So it's now a very powerful technique. And then I just look at the World Economic Forum, the prediction of the top 10 emerging technologies. Of course, you have predictions in structural battery, where the cars will have to use this structure. It's the car itself as a part of the battery, osmotic power systems from water purification, advanced nuclear technology, engineered living therapeutics, which will be part of molecular biology, and the neurodegenerative diseases, autonomous biochemical sensing. Green nitrogen fixation is a big problem because that uses a lot of energy. And then we have nanoscience, collaborative sensing, and generative watermarking to tell whether using AI for a particular write-up or not. So they can determine if you use AI. So the students, if you use AI, this will be a big check on whether you are using your mind or not. So despite our success in science, so something seems to be lacking. And I thought of being in the Philippines. It's such a poor country that what can I do for the Philippines? So I started working with the ITAS and had a conservation project with them and a livelihood project. And then I became involved in Future Earth, which is a network of scientists and engineers for global it is a global initiative for sustainability. And it's because the reason we thought about this, because according to the Ecological Footprint Network, we have been on ecological deficit since 1960s. So our biocapacity has been going down. Our ecological footprint has been going up. And our deficit is now minus 0.8 hectares per person. So it's worrying. That's why we decided to form Future Earth Philippines as a remedy for us to get together, to work together for sustainability and resilience. And I think the other nations should also start worrying about this, because those in the red are also ecologically deficit. Only those in green have not yet reached the deficit stage. And that I like this diagram of the SDG, Sustainable Development Goals, because it tells us that the foundation for sustainability should be a healthy biosphere. Life below land, life below water, clean water and sanitation, and climate action. And then, if we have this healthy biosphere, we can build a good society, a stable society, and perhaps develop a very good economy. So for as the Asia and the world stands at the critical juncture, we are confronted with climate change, biodiversity loss, food, water, energy, and health insecurity, and mounting urban pressures, which are all problems in the Philippines. Asia and the world must mobilize every tool available to achieve sustainable, inclusive development. And the heart of this transformation is science and technology. And at the forefront of this movement, you are at the forefront of this movement, the youth, because you will be inheriting the earth. We will disappear after some time. I'm now 83 years old, so not much is left. Uh, but the youth of the day can still do very much. So working together for the world we want and using science, technology, and innovation, let's collaborate. Use your curiosity, scientific thinking, innovation, and then be honest, ethical, and always consider the values, both professionalism and even the cultural values. Thank you very much.
Thank you very much, Professor Dr. Cruz, for inspiring and thought-provoking lecture. So we have learned a lot about her journey to become a famous scientist and travel around the world, and then apply her knowledge and help like poor and rural people saving the earth. So on behalf of all participants, we express our heartfelt thanks for our invaluable insights. Thank you very much again.
Thank you very much.
Okay, I think it's time for questions. So we have about 17 minutes for questions. Okay, so please. Yeah, we don't
Hello,
have
my
any-
name is Thanadesh from Thailand. My question is, if the chronotoxin have like a species have many of them, can we extract the antibodies the from their self, and like can we mimic them? Can we create them to use in the human to prevent a chronotoxin in venoms? Yeah.
Well, we didn't. The cone snail stings are quite rare. I mean, since they started recording these, only 144 have been recorded, and about most of the fatal ones are from Conus geographus. So I don't think anybody will invest in making. Well, nobody will be able to make money out of the antibodies. It's more business, the economy of it. And plus, since these are small peptides, we don't think they are very antigenic. Although some have read of where they inject this in chicken and they collect the eggs for production of the antibodies, but these are not humanized antibodies.
Thank you.
Professor Cruz, really appreciate for your
Thank you.
lecture. I'm Zhang Lequan from China. So as you're mentioning the lecture and a retrospect paper you published and with your team, you mentioned that one of your students, Craig Clark, used a method,
Yes.
the intracranial injection, and
Yes.
this method can better distinguish the function of the canotoxin. So I wonder that why this injection method can be more effective than the IP injection. Is that mainly due to the blood-brain barrier or other factors?
Well, we have different ion channels in the periphery and maybe a lot of more different channels in the brain where they are acting very specifically. They have very high specificity, although they have very high affinity, but very high specificity also. So what evolved for against fish may not be okay for humans, may not affect, but may be able to interact with. There are many different subtypes of ion channels and receptors, and they react only with a specific subtype. So which that are not present in our periphery.
Okay, thanks, Professor.
Thank you.
Okay, my name is Indy and I'm from Thailand. Chronotoxins have specificity for the ion channels and receptors. Do you think their precision could be harnessed to create next generation modulators to avoid the side effect of painkillers and use them to target the dysfunctional neural circuits with a near surgical accuracy?
Yes, I think, but for example, in the omega conotoxin, we thought it will be a very good one for pain alleviation. But in some people, they have observed some side effects that are serious. And I think the new technique of constellation pharmacology can tell you what the other effects will be on the different cell types. And that should be used more in working with, say, natural products and these peptides and so on. That will offer a very good tool.
All right, thank you.
Thank you. So, no more?
My name is Archana from India. My question is, how can we estimate the multi-target effect of conotoxology?
Again?
How can we estimate the multi-target effect of conotoxology?
Let me see. How can we...
Multi-target effect.
Oh, multi-target effect. The conotoxins are very specific. So there are many, many different peptides, but we can use a combination, of conotoxins to get the desired phenotypic changes that we want to have. Because they are very specific, they're highly specific, and high affinity. So we thought that they would give us something that will not have any side effect. Unlike the natural products, which are relatively small, their affinity is not that high for different ion channels and receptors.
Thank you.
Hello, I am Tita Hamdragun Shefrik from Thailand, and I was wondering, in the chemical characterization method, you said that you used the chromatography to study
Yes,
the component of the toxin. Is there any extraction method or separation method that can be used to study? Yeah.
Well, now we just use the HPLC for separating the whole venom because it's very high resolution and really offers a good separating technique. And there are different kinds too that one can use.
Okay, thank you so much.
And it's convenient now because you have automated sequencer, automated analysis, and synthesizers, and so on. So it's much easier, I guess, to work with peptides.
Hello, my name is Chayut Wanasinto from Thailand. My questions here is about the mistakes where when extract the toxins and then inject it to the mouse. Is there like any mistakes happens during the process of that?
Yeah, I was telling you, we thought a particular thing that we isolated is the peptide that was giving us the activity from the native venom, but it turned out to be only what they call a contaminant of the peptide that's truly active. So one has to be careful. One has to synthesize and prove that it really has the activity before concluding that this is the toxin.
Ah, okay. Thank you, Miss.
Hi, my name's Kanako. I came from Japan, and you talked about importing machines for experiments. I just wanted to know what the funding system was behind that. Where did you get that money? Because I'm guessing that's pretty expensive to do.
When I was starting, at that time, the International Foundation for Science, based in Sweden, still offered grants for natural products research because Jack Cannon, Australia, was the one who's supporting the people from Southeast Asia. But now I think the International Foundation for Science is focused more on Africa. But there may be like IDRC of Canada, and maybe you can collaborate with somebody in the US
Interesting. Thank you.
or Europe. But now maybe it's probably difficult because Trump is around.
Okay, still one more.
Hello, I am Thanadesh again from Thailand. I have learned in my zoology lecture that the chronotoxin have something related with a morphine addiction. You may have mentioned that the intra brain injection is possible in the chronotoxin. My question is this possible to use a chronotoxin as a morphine addiction prevention?
Well, probably constellation pharmacology can show you first what it may do instead of using whole organisms. For constellation, you can apply it, a set of ligands to cells, and then look at them individually.
Thank you very much.
Okay, any questions? Okay, so if not, so once again, let us thank Professor Dr. Cruz for very exceptional lectures.